Hello, everyone, and welcome. I am Michael Hall. I will be talking about a label-free method for detection of SARS-CoV-2 spike protein using an electrochemical biosensor. This is my thesis project, and this is going to be a quick run through of a proposal for this project that I've already presented for my mentors. So to start off, we're going to be talking about severe acute respiratory syndrome, coronavirus 2. This is SARS-CoV-2. It's been hard to avoid this topic by any means. It is responsible for the pandemic that we are currently in and have been in for about a year and a half now. It's an RNA virus, which is interesting, and it consists of a single strand positive sense RNA of about 30 kilobases. It is similar to many coronaviruses. In fact, it shares a 82% sequence identity with SARS-CoV and MERS-CoV, but even more of a sequence identity is similar for these essential enzymes and proteins. These would be proteins such as the nucleocapsid or the matrix or membrane protein, the envelope protein, or the protein we'll be discussing a lot more today, the spike protein. The virion is about 65 to 125 nanometers in diameter, and that is not counting the spike protrusions, the spike proteins that stick out of it. The spike protein has a receptor binding domain, which is the RBD, and this binds to the ACE2 of the host cells, and this allows for the start of the infection of the cells. The angiotensin converting enzyme 2 is the ACE2. So it plays a role in the renin angiotensin aldosterone system. It actually is the second ACE2, or the ACE protein, and it breaks down angiotensinogen 2 into angiotensinogen 1 through 7. This system is involved with inflammation and healing when is required, but also in maintaining a blood pressure. So not rising, not lowering, just kind of maintenance on that. However, it has a high affinity complex with the spike RBD, as it is a receptor itself. And this complex is very strong, and this is based off of the many hydrogen bonds, so 13 hydrogen bonds and 2 salt bridges that form between the ACE2 and the RBD. Where one might assume that when it is bound, it may not be able to perform its job as well. In fact, it enhances the cleaving once this complex is formed, and the ACE2 will now cleave molecules more than it did prior to being bound to the spike RBD, but with less specificity. These two proteins that I have been discussing, the spike RBD and the ACE2, are going to be used in an electrochemical biosensor for my thesis project. At a base level, it is a change in electric signal through chemical reactions of binding to sense a biotic material. So this could be DNA, RNA, proteins, just molecules. As long as the ligand that is detecting for the analyte can be bound in some way to that analyte. One important thing here is that a lot of detection methods will use a modifying of the analyte to have a fluorophore. And once the binding happens, the fluorophore is released and it will create some kind of color or a light. And this does not require changing of the target molecule, the analyte. It will be connected to a circuit, and the circuit will have a voltage at constant voltage. And with a constant voltage, you'll have a current. The current is going to change based off of the binding of the analyte to the ligand. In the research that we did on this topic, we found other electrochemical biosensors made for the purpose of hopefully detecting SARS-CoV-2 in some way or the other. And in that time, we found some using graphene, like in the top left picture of this slide. They used a linker, which is P-base in this case, to bind the antibody of the whole SARS-CoV-2 to the graphene. And then they released the SARS-CoV-2. In this case, they used all synthetic material. They created their own SARS-CoV-2. They created their own antibodies and their own graphene. In the top right, they used a different method by using a gold-coated carbon nanotubule with a linker of thiol bound to a cDNA strand that will complement the RNA of the SARS-CoV-2 virus. They would be able to detect the RNA in a sample. And in the bottom left, there was some research being done on binding ACE2 to a nanoparticle, which was important to this research as it hadn't been done for a electrochemical biosensor as we are intending to do. So we wanted to make sure that it was possible. And in this case, they were not using it for electrochemical biosensor, but they were using it to hopefully be able to create like recombinant ACE2, and they want to be able to like flush it into the system of a host, and in the hopes that it could, these ACE2s, these free-floating ACE2s could bind to the spike RBD of the virus instead of it binding to the anchored ACE2s of the cells of the host. And they wanted to just see what the affinity was for this. So that was what this part was about. And we found that was very useful information for us to be able to do this ourselves. Our hypothesis is that we would be able to fabricate and test this electrochemical biosensor that is being made in Dr. Kumar's lab for finding and interrogating samples of SARS-CoV-2 spike protein via the direct binding of our ligand, the ACE2. Our fabrication method is to use drop casting of a multi-walled carbon nanotubes in methanol. And as it will be just a very small drop of 1 microliter onto a interdigital electrode, and it will dry. And as it dries, it will create this layer, a top layer of the carbon nanotubules on the electrode. Drop casting will be done again of 1 microliter of 250 micrograms per mil of the ACE2 ligand onto the device. This will sit overnight so that the pi-pi bonds will form between those two. Once that is done, we will do a wash to remove any of the ACE2 that did not form fully and has a weaker bond. At both stages of the pristine carbon nanotubes dried onto the device, the electrode, as well as the ACE2 ligands that have been bound, we will observe with physical characteristics of this under the scanning electron microscope, as well as the electrical characteristics by inserting the device into the circuit that we'll be using for our actual experiment. We will have similar pictures as this and graphs as this. So in the right picture, we have a 5 micrometer image from a scanning electron microscope of carbon nanotubes. This is not connected or attached to ACE2, but it is attached to a different type of protein. You can see there are darker spots here of this image, and the lighter are the proteins that are bound. It creates a non-pristine coverage. In the left-hand side, you can see that there is a graph that shows pristine graphene current over voltage and a change as every component is added to this. So they have an intermediate molecule of the P base attached, and that's in red. And then when they have the P base and the SARS-CoV-2 antibody attached, it is in blue. And it is changing as these different molecules are attached and bound. So once we begin the detection of the analyte, which is the main experiment here, we will be starting by entering the device into the circuit and probably start our first control, which would be the running buffer of PBS over the biosensor. There should be no change in current at this point because there will be no analytes added, so no binding will occur. However, as you add a range of analyte concentrations from 1.2 nanomolar to 1.2 micromolar, we will see changes in the current, spikes in the current. And this current density is going to be able to determine the concentrations of the analyte in the solution. Once that has been done, we will be able to do a dilution series to find the lower range of detection that we can have with this device. This is important because if it is able to detect at very low concentrations, it could be very helpful in determining if a sample has COVID-19 or SARS-CoV-2 spike proteins are in the solution, in the sample. Another device will be used eventually that does not have a mobilized ACE2 on it, and we will then run the range of analyte concentrations onto that device to see if there is any binding that is occurring that does not need the ligand. And that could be problematic, as if any spike protein is binding directly to the carbon rather than directly to the ligand, it could mess up or prove that there is no real detection being observed. Then we will move on to interference assays, and this will be done by using competition of other substances, such as in a human tissue culture extract that will be grown in Dr. Hunter's lab, and it will be ground up and mixed with known concentrations of the analyte, and that may interfere in some way with the detection accuracy. This will be as well with the universal transfer media, because it could be very convenient to have the sample in a universal transfer media and then be directly taken out. And if it is still accurate in our detection method, that would be very useful. Another test we're going to be running is with the spike proteins of variants to detect if the affinity for SARS-CoV-2 spike RBD is enough to have a very accurate concentration reading, or if we can easily get a concentration reading with different spike variants. Because we would want to be able to say that if we got a sample and they had SARS-CoV-1, it would not detect at the same rate as somebody who has a sample with SARS-CoV-2, since this was made for SARS-CoV-2. But we could easily change one or two parts to be able to then make a sensor for that if needed. And that is all. I have my references here, and thank you for watching.